Unit-1 Carbohydrates MFN 002 Nutritional Biochemistry First Year Master of Science, Food Nutrition, MSCDFSM, Revised Master's Degree Programs, School of Continuing Education, SOCE, IGNOU Audiobooks #IGNOU Audiobooks Indira Gandhi National Open University 1.1 Introduction. Nutrition is a relatively new science that evolved from disciplines such as chemistry and physiology. Thus, a student who intends to know and practice nutrition and dietetics should not only undertake intense learning and training on the different techniques that relate nutrition with chemistry, but they should also understand how nutritional processes are merged with chemistry. Further, it is important to have a clear idea of the chemistry of nutrients for a better understanding of nutrition. Do you have such knowledge? If the answer is no, then it is absolutely necessary that you should learn intensely this unit, and you will find that the contents provided in it will serve as the foundation for the other courses such as the Advanced Nutrition, Principles of Food Science and Public Nutrition courses. If your answer is yes, then too you must go through the contents of this unit to assess if you have the correct understanding of the subject and to brush up your previous knowledge, and at the same time, to gather any new knowledge that you may discover during study. This unit will provide you with a broad introduction to metabolic and nutritional aspects of biochemistry. The major focus is on the structure, function and metabolism of carbohydrates. Objectives: after studying this unit, you will be able to understand the meaning of nutritional biochemistry and discuss its evolution, classify carbohydrates, and describe the chemical properties, viz. mono, oligo and polysaccharides. 1.2 Introduction to Nutritional Biochemistry. Being a student of dietetics, a clear understanding of the term nutritional biochemistry, as well as its development, is pertinent to your knowledge. Equally important is the applicability of nutritional techniques at the advanced level. This subsection, therefore, intends to make the meaning of nutritional biochemistry clear to you and inform you about the evolution of this science over the centuries. Let us get started then. 1.2.1 Meaning and Importance of Nutritional Biochemistry. Nutrition is a science that cuts across several scientific disciplines, of which one is biochemistry. It is the science that relates food to the functioning of the living organism. Nutrition includes the intake of food, digestion and absorption of food, transport and assimilation of nutrients, metabolism, liberation of energy, and elimination of wastes. It, in fact, encompasses all the synthesis essential for growth, maintenance and reproduction. Biochemistry, as per definition, is the chemistry of living organisms that covers all the chemical reactions occurring in our body. Biochemistry explores the functioning of living organisms from a molecular and cellular perspective. As implied by the course, it emphasizes the problems of nutrition and the mechanisms of biochemistry. The reason for this is quite understandable, as nutrients are basically chemicals and our body is a chemical machine composed and built up of chemicals. Therefore, the biological function of a nutrient is readily explained in terms of its chemical interaction at the molecular level with the body's endogenous chemicals. A good understanding of the biochemical basis of nutrient function and of the consequence of nutrient deficiency or excess is important in the clinical and laboratory diagnosis of nutritional diseases, clinical management of the same, and in the control of endemic nutritional inadequacies. You, as a student of dietetics, should realize the importance of biochemistry in order to a. Understand nutrition properly, and b. Utilize biochemical tools in solving nutrition-related health problems. With this basic understanding of biochemistry and its relation with nutrition, let us move on to understanding the development of nutritional biochemistry as a discipline and contemporary interests in this area. 1.2.2 Development of Nutritional Biochemistry. Nutrition, in general, and particularly nutritional biochemistry, has traveled a long way since 1926, when nutrition was first recognized as an independent field of study with the appointment of Mary Swartz Rose as Professor of Nutrition at Columbia University. This was actually the culmination of a developmental period stemming from Antoine Lavoisier's experiments about 200 years back that formed the basis for the studies on respiratory exchange and calorimetry, the beginning of a new science, nutrition. About 100 years elapsed before carbohydrates, fats and proteins were identified as the source of energy for animal body. By the end of the 19th century, the necessity for certain minerals in the diet was established. But it was not until the decade between 1930 and 1940 that the majority of the vitamins were identified, isolated from different foods, synthesized in the laboratory, and received serious attention in order to understand their involvement in various biochemical reactions. The contemporary interests in nutritional biochemistry are highlighted next. 1.2.3 Contemporary interests in nutritional biochemistry. As newer techniques in biochemistry have emerged from time to time, these were of immense help in the understanding of utilization of different nutrients by the body. Biochemical bases of some of the dreaded nutritional diseases of the past, such as beriberi, scurvy, pellagra and pernicious anemia, are well understood. Likewise, problems such as protein energy malnutrition, xerophthalmia, iron deficiency anemia, iodine deficiency disorders, though well understood, are still prevalent. Added to these are contemporary problems like diabetes and cardiovascular diseases. Reliable biochemical indicators are also now available to detect the prevalence of subclinical deficiency of many nutrients in the population. Once the generation of free radicals in the body was correlated with the development of different fatal or crippling diseases, the importance of antioxidants increased many folds. In recent times, some nutrients such as vitamin E or selenium are getting special attention from research point of view, as these are potent antioxidants. Recent biochemical studies have not only revealed the importance of polyunsaturated fatty acids in maintaining the normal lipid profile, but these also went further to prove that a desirable ratio of N3, N6 fatty acids is essential for preventing dyslipidemia, a condition in which an abnormal amounts of lipids and lipoproteins accumulate in the blood. Importance of nutritional interactions is now being realized. Some nutrients are antagonistic to each other, whereas others act synergistically. Examples of uniquely related nutrients include folate and vitamin B12, vitamin E and polyunsaturated fatty acids, vitamin D and calcium, zinc and copper, etc. Biochemical research has revealed that certain drugs such as omeprazole, lovastatin, allopurinol, thiouracil and others influence nutrient metabolism. Not only nutrients, but the importance of non-nutrients such as dietary fiber for healthy living is also understood. Very recently, a group of compounds known as phytoestrogens has created great interest among nutritional biochemists. These substances are considered to protect cancer, besides having other positive roles. As a consequence, many popular, as well as uncommon foods and beverages are constantly under study to explore the presence of these compounds, as well as to observe their biochemical effects. Tea and soybean are two good examples. Nutraceuticals are gaining popularity at an exponential rate. These are classified as foods that provide medical or health benefits. Spirulina is already available in the Indian market besides other countries with high claims. Few uncommon edible oils such as flaxseed oil, primrose oil etc., which are already in use in the Western countries for their apparent beneficial effects, may at any time enter the market in India. Though many of these preparations have proven positive effects on health, a more in-depth biochemical research will definitely help to know about these new generation nutritional substances. The discussion above presented a good review about nutritional biochemistry, its development and scope in modern times. With this basic understanding, we shall move on to the study of the chemistry of important food constituents, namely carbohydrates, proteins, fats, etc. In this unit, we shall focus on carbohydrates. 1.3 Chemistry of carbohydrates. This is absolutely essential for you, as a student of dietetics, to have a clear concept on the nature and physico-chemical properties of different nutrients. After studying this subsection, you will be able to know the following important aspects of carbohydrates: their chemical nature, their classification, and their physico-chemical properties. Let us start by first getting to know what are carbohydrates. You would realize that the family name ending "ose" indicates a carbohydrate, for e.g. glucose, fructose, sucrose, etc. Carbohydrates are basically polyhydroxyaldehydes or ketones and their derivatives. What do we mean by polyhydroxyaldehydes or ketones? Go ahead, read on and you will find the answer to this question soon. Carbohydrates, as you may already know, are found abundantly in both animal and plant tissues in different forms. While plants can produce carbohydrates by photosynthesis, animal cells cannot but are dependent on food for these important sources of energy. Some carbohydrates have highly specific functions in the body. Let us learn about these different carbohydrates. Carbohydrates are classified into three major groups. 1. Monosaccharides. 2. Oligosaccharides. 3. Polysaccharides. Monosaccharides and oligosaccharides are also referred to as sugars. Let us get to know about monosaccharides first. 1.4 Monosaccharides. Monosaccharides consist of a single polyhydroxyaldehyde or ketone unit and are commonly known as simple sugars. These cannot be hydrolyzed into simpler forms. The general formula for monosaccharides is CH2O(N), where N is 3 to 7. In biological materials, monosaccharides with 5 and 6 carbon atoms are most common. All the carbon atoms of the monosaccharide contain one hydroxyl group, O, except one that contains a carbonyl oxygen, as in CHO or greater than C equals O. If it is present at the terminal position, the monosaccharide is an aldehyde derivative and is called as an aldose, sugar position, e.g. glucose. On the other hand, if it is present in any other position, the monosaccharide is a ketone derivative that is known as a ketose, e.g. fructose. The carbon atoms are numbered as shown in the figure 1.1. C1 atom is an aldehyde functional group and C2 atom is a ketone functional group as highlighted in figure 1.1. So now you know how carbohydrates get classified as polyhydroxyaldehyde or ketone. Figure 1.1. Numbering of carbon atoms in monosaccharides. Look at the screen. Monosaccharides can further be divided on the basis of the number of carbon atoms they possess. Table 1.1 will be helpful to understand how the monosaccharides are classified, considering the number of carbon atoms in the molecule, as well as the nature of the carbonyl oxygen with specific examples. The number of carbon atoms in an aldose or ketose may be specified by triose, tetrose, pentose, hexose or heptose. For example, glucose with 6 carbon atoms is an aldohexose and fructose 2 with 6 carbon atoms is a ketohexose. Table 1.1. Different types of monosaccharides found in foods. Look at the screen. So far we have learnt about the different types of monosaccharides. It is also important to understand that monosaccharides exist as isomers. We shall understand this concept better by learning next about the isomers of monosaccharides. 1.4.1 Isomerism of monosaccharides. What do we mean by the term isomer? Existence of different compounds having same molecular form but different structural forms are isomers. Monosaccharides exhibit a variety of isomerism such as stereoisomerism, optical isomerism, aldose-ketose isomerism, etc. These isomerisms are described as follows. Aldose-ketose isomerism. We have already seen that in a monosaccharide either an aldehyde, CHO, or a ketone, greater than C equals O, group is present. The former is called as aldose while the latter is known as ketose. Glucose and fructose both have the formula C6H12O6, but glucose is an aldohexose, aldehyde-bearing hexose, and fructose is a ketohexose, ketone-bearing hexose, so they are isomers to one another. Figure 1.2 graphically represents this isomerism. Look at the screen. Figure 1.2. Aldose-ketose isomerism. Stereoisomerism. Stereoisomerism occurs when the same compound, due to different spatial arrangement of the groups attached to its asymmetric carbon atom, exists in more than one form. First, let us understand what do we mean by an asymmetric carbon atom. When any carbon atom of a compound is attached with 4 different groups or atoms, it is called as an asymmetric carbon atom. Look at figure 1.3. Why is the C2 atom the determining factor in this configuration? The carbon atom C-2, marked here with an asterisk, is unique since it has 4 different groups, OH, CHO, H, CH2OH, attached and is therefore an asymmetric carbon. The C2 atom is also called the chiral carbon atom. According to this observation, monosaccharides may have 2 different forms, namely D sugar and L sugar. depending on their relation to the direction of the O group on the number 2 carbon atom as illustrated in figure 1.3. The D form will have the O group next to the bottom carbon atom, primary alcohol group, on the right side, while the L form will have it on the left side. The simplest 3 carbon atoms naturally occurring glyceraldehydes, as indicated in figure 1.3, lack a plane of symmetry and exist as a pair of enantiomers, stereoisomers that are mirror image of one another, D and L forms, which are the mirror images of each other. In this context, carbohydrates are chiral molecules since they have carbon atoms carrying 4 different groups. The majority of the monosaccharides occurring in mammalian metabolism are of D configuration. Look at the screen figure 1.3. Structural isomerism of glyceraldehyde, another type of stereoisomerism known as epimerism, occurs with respect to a single asymmetric carbon atom of a monosaccharide possessing more than one asymmetric carbon atom. In figure 1.4, there are 3 6-carbon hexose sugars. 4 carbon atoms, C2, C-3, C4 and C-5, are chiral atoms. These are structural isomers, i.e. they have the same molecular formula of C6H12O6, but different structural formula, and consequently they differ in their physical and chemical properties. Actually, these isomers are formed as a result of interchange of the O and H on carbon atoms 2, 3 and 4 of glucose. Glucose and galactose differ in the configuration of a single carbon atom 4, while glucose and mannose differ in C-atom No. 2. As indicated in figure 1.4. Compounds that differ in this manner are called as epimers. In general, compounds with N chiral carbon atoms have a maximum of 2 N possible stereoisomers and half that many pair of enantiomers. For example, glucose and aldohexose has 4 chiral atoms and a total of 24 equals 16 possible stereoisomers, 8 pairs of enantiomers. Look at the screen figure 1.4. Epimerism of glucose, galactose and mannose. Optical isomerism. A compound is said to have optical activity when it rotates the plane of vibration of the rays of polarized light passing through it. This in fact results due to the presence of asymmetric carbon atoms, carbon atom in a compound attached to 4 different groups or atoms in the molecule. 2 compounds having a similar molecular formula may have different optical activity. When an optically active substance rotates the plane of polarized light in a clockwise direction, it is called as dextrorotatory or D isomer of the substance, and when it rotates the plane of polarized light in the anticlockwise direction, it is levorotatory or L isomer of the substance. The D and L isomers are also expressed as plus and respectively. If you look at figure 1.3, the 2 forms of glyceraldehydes, D and L forms, rotate plane of polarized light in the opposite direction by the same amount. As it turns out, D-glyceraldehyde rotates the plane of polarized light to the right and is therefore dextrorotatory, labeled as plus. So far we have learnt about the different isomers of monosaccharides. You would notice that certain cyclic forms of monosaccharides are also present. These are explained next. Cyclic form of monosaccharides: aldohexoses, e.g. glucose and galactose, ketohexoses, e.g. fructose, and aldopentoses, e.g. ribose, in solution undergo cyclization when treated with equivalent amounts of alcohol and form a hemiacetal or a hemicatal in the aldose or ketose, respectively, linking the carbonyl carbon atom with the other carbon atom by SOCE linkage. This results in ring formation, that is either a 6-membered ring with 1 oxygen atom for aldohexoses or 5-membered ring with 1 oxygen atom for ketohexoses. The former is known as paranose and the latter as furanose, as illustrated in figure 1.5. Look at the screen figure 1.5. Ring structure of monosaccharides. Glucose, like all other monosaccharides, exists in 2 forms, the open chain form and the ring form, as illustrated in figure 1.6. The ring form has an additional asymmetric carbon atom called the anomeric carbon atom. The asymmetry of C-1 makes possible the 2 ring forms, alpha and beta, with different optical rotations, mutarotation. Mutarotation is the process whereby the configuration of an anomeric carbon converts from alpha and beta and vice versa. The alpha form has the O group on the reverse side in comparison to beta form at C-1, as highlighted in figure 1.6. This ring form of sugars, hemiacetals, containing an asymmetric carbon atom can exist in alpha and beta anomers. Anomers are cyclic sugars that differ only in position of substituents at the hemiacetal carbon. The alpha form has the O group on the opposite side from the CH2OH. The beta form has the O group on the same side as the CH2OH group, as is evident in figures 1.6 and 1.7. Glucose in its pyranose form is called as alpha-D-glucopyranose. Fructose in its furanose form is called alpha-D-fructifuranose as shown in figure 1.8. Figure 1.7, alpha and beta anomers of D-glucose. Figure 1.8, alpha and beta anomers of fructose. Look at the screen. We have observed that monosaccharides can exist in different forms. Why do you think the study of these forms was important? The structure or the form in which monosaccharide is present influences the properties of monosaccharides. We shall learn about the properties of monosaccharides after answering the Check Your Progress exercise 1. 1.4.2 Properties of monosaccharides. Monosaccharides form water-soluble, colorless, odorless, sweet crystals and show specific reactions of the aldehyde or ketone and hydroxyl groups, which help to identify these compounds. Some of the important reactions of monosaccharides are elucidated here. Esterification. As monosaccharides possess hydroxyl groups, O, they form esters with acids. Phosphoric acid esters of glucose and fructose have a great significance in nutritional biochemistry, as these are the metabolic intermediates of glucose or fructose. For example, fructose 1,6-bisphosphate. We will learn more about this later in Unit 6. Figure 1.9, esterification of glucose to glucose 6-phosphate. Oxidation reactions. Mild oxidizing agents such as sodium hypoiodide, NaOI, and bromine water oxidize aldoses to aldonic acids when CHO, aldehyde group, of the aldose is converted to COOH, carboxylic group. Glucose is oxidized to gluconic acid as shown in figure 1.10. Figure 1.10, oxidation of glucose to gluconic acid. Look at the screen. On the other hand, strong oxidizing agents, such as nitric acid, oxidizes aldose to glucaric acid, aldaric acid. See figure 1.11 for reaction. Here both CHO and CH2O are oxidized to COO. Glucose is oxidized to glucaric acid. As ketoses do not undergo oxidation, this reaction may be used to distinguish an aldose from a ketose. Figure 1.11, nitric acid. Oxidation of glucose to glucaric acid. Dilute hydrochloric acid, HCl, oxidizes only the terminal CH2OH group with the formation of uronic acid. Glucose is thus oxidized to glucuronic acid. Look at the screen. Figure 1.12, oxidation of glucose to glucuronic acid. This is the basis of Felling's test and explains the term reducing sugar. In Felling's test, the free sugar group, aldehyde or ketone, reduces the Cu2+ cupric ions in an alkaline environment to form copper-1 oxide, cuprous oxide, and the sugar is itself oxidized to a carboxylic acid, mixture of sugar acids. The copper-1 oxide is red and precipitates. Carbohydrates that react in basic solution with oxidizing agents are classified as reducing sugars. In basic solution, all monosaccharides, whether aldoses or ketoses, are reducing sugars. Simple sugars can act as reducing agents because the aldehyde or ketone group is readily oxidized to carboxylic acid. Reduction reaction. Aldoses and ketoses are reduced to the corresponding polyhydroxy alcohols by sodium borohydride, sodium amalgam, etc. So, in this reaction, as given in figure 1.13, glucose is reduced to glucitol, sorbitol. Polyhydric alcohols are also called sugar alcohols and are the hydrogenated forms of the aldoses or ketoses. Glucitol, also known as sorbitol, has the same linear structure as the chain form of glucose, but the aldehyde, CHO, group is replaced with ACH2OH group at C-1. Sugar alcohols have about half the calories of sugars and are frequently used in low-calorie or sugar-free products. Look at the screen figure 1.13. Reduction reaction of glucose, osazone reaction when a monosaccharide, or a reducing disaccharide, is heated with phenylhydrazine, a mild oxidizing agent, in acetic acid. The carbonyl group of the monosaccharide reacts with phenylhydrazine to form a compound known as osazone. Osazones have characteristic yellow or orange microscopic crystals of specific melting points. This reaction can thus be utilized to identify different sugars. Figure 1.14. Formation of osazone. Furfural formation. When treated with concentrated mineral acids, monosaccharides are transformed to a class of compounds known as furfurals. We have studied about monosaccharides in last section. Next, let us get to know about the oligosaccharides, their structure and chemical properties. 1.5 Oligosaccharides. Oligosaccharides contain 2 to 6 monosaccharide units joined by a linkage known as glycosidic acetyl linkage. On the basis of number of monosaccharides present, these are divided into disaccharides, trisaccharides, etc. Disaccharides are the most important compounds in this group. These consist of 2 monosaccharide units, molecules joined by a glycosidic linkage. Depending on the nature of monosaccharides present, different disaccharides are formed as highlighted in the Table 1.2. Table 1.2. Constituents of disaccharides. Disaccharides. Monosaccharides present maltose glucose, 2 molecules, lactose glucose, galactose sucrose glucose, fructose. The glycosidic linkage or bond is formed through a reaction between AO group on the anomeric carbon of one unit and AO group contributed by the other unit. In sucrose, the second O group is also contributed by an anomeric carbon atom, but in maltose and lactose, it is contributed by carbon atom 4. The configuration of the disaccharide glycosidic linkage depends on the anomeric form of the unit contributing the anomeric carbon atom. Thus exists alpha 1,2 glycosidic linkage in sucrose, carbon 1 of glucose in the alpha orientation to carbon 2 in the beta form of fructose, alpha 1,4 in maltose, 2 glucose units joined from C1 to C4, and beta 1,4 lactose. The figures in the brackets indicate the position of the glycosidic linkage between the 2 monosaccharide units. The configuration of the glycosidic linkage affects the chemical properties of the disaccharide. Disaccharides with free anomeric carbon atoms like monosaccharides exhibit the characteristic reduction reaction of carbonyl group with Felling's reagent and Benedict's reagents, an alkaline solution containing a cupric citrate complex ion. Sugars which reduce these reagents are called as reducing sugars. Sucrose, having no free anomeric carbon atom, does not respond to this reaction. Linkage of the disaccharides are shown in Figure 1.15. Figure 1.15. Structure of disaccharides. Besides disaccharides, some trisaccharides, possessing 3 monosaccharide units, are also freely available in nature. One important member of this group is raffinose, which is made up of the derivatives of galactose, glucose and fructose. Disaccharides consist of characteristic anomeric forms of monosaccharide units. Sucrose consists of alpha-D-glucose and beta-D-fructose, maltose consists of 2 alpha-D-glucose units, while lactose consists of beta-D-galactose and alpha or beta-D-glucose units as shown in Figure 1.15. Disaccharide molecules in which the second hexose unit has a free anomeric carbon atom may undergo mutarotation, and the free anomeric carbon is capable of existing in alpha and beta forms. Sucrose, however, is an exception as it has no free anomeric carbon. Next, let us learn about polysaccharides. 1.6 Polysaccharides. Polysaccharides are produced when many monosaccharide units are joined together by glycosidic linkage. Chemically, polysaccharides are of 2 types, A. Homopolysaccharides or homoglycans which possess only one type of monosaccharides, such as starch, cellulose etc., and B. Heteropolysaccharides or heteroglycans which are formed by more than one type of monosaccharides, such as hyaluronic acid, heparin. Table 1.3 lists the commonly occurring polysaccharides of different types. Table 1.3. constituents of common polysaccharides. Look at the screen. Let us move on to studying each of these polysaccharides, their components and structure. We shall begin our discussion with the most commonly known of these, starch. Starch is a plant polysaccharide synthesized by the plant through photosynthesis and stored mainly in grains, legumes, roots and tubers. Its molecular formula is C6H10O5N. Starch consists of two forms, amylose and amylopectin. Normally, 65 to 85% of the starch is amylopectin and only 15 to 35% is amylose. Amylose has a straight chain structure formed by 250 to 300 glucose residues linked together by alpha-1,4 glycosidic linkage as shown in figure 1.16. In aqueous medium, it assumes a folded conformation. Amylopectin, on the other hand, is a highly branched polymer of glucose. In amylopectin molecule, branching occurs at intervals of 24 to 30 glucose residues. Glucose units of the main chain are joined by alpha-1,4 glycosidic linkage, similar to amylose, and the glucose units at the branch are joined by alpha-1,6 glycosidic linkages to the main chain, figure 1.16. Amylopectin generally has about 3,000–6,000 glucose residues. Figure 1.16, schematic diagram of amylose and amylopectin. Starch is not soluble in water and forms colloid in aqueous medium called as micelles. Iodine reacts with both amylose and amylopectin, but the former produces a complex that gives an intense blue color and masks the violet color of the complex formed by the latter. As a result, we observe a blue color when starch reacts with iodine. This color complex dissociates on heating and reforms again after cooling. Starch is a non-reducing substance, as except one carbonyl group, all other carbonyl groups are involved in glycosidic bond formation. It may be hydrolyzed by boiling with hydrochloric acid or by the action of enzyme amylase ultimately to free glucose. Now, we move on to cellulose, most abundant polymer of glucose, found only in plants. Cellulose, it is the main constituent of plant cell walls and the most common and abundant of the D-glucose polymers. This does not occur in the animal body. It is a homopolymer of glucose-like starch, except the linkages joining the glucose units are beta-1,4 rather than alpha-1,4. Hence, cellulose is made up of beta-glucose molecules linked by beta-1,4 glycosidic linkage as shown in figure 1.17. Due to the difference in the chemical structure, it is not acted upon by the enzyme amylase of the digestive juice. Strong hydrochloric acid hydrolyzes cellulose to glucose. Figure 1.17, beta-1,4 glycosidic linkage in cellulose. Glycogen is the storage polysaccharide found in the muscle and liver of animals and humans. It is a branched polymer having about 8 to 10 glucose units in each branch. Like amylopectin, its straight chains are formed by alpha-1,4 glycosidic linkages and alpha-1,6 glycosidic linkage exists at branch points. Thus, the chemical structure of glycogen and amylopectin are similar except that the former is more branched. Each glycogen molecule may contain 5,000 to 10,000 glucose units. It is non-reducing, readily soluble in water and gives a red color with iodine. Now, we move on to the not so commonly known polysaccharides, dextrins and inulin. Dextrins are also polymers of D-glucose held by alpha-1,4 glycosidic linkages. Dextrins are, in fact, formed due to partial hydrolysis of starch by enzymes such as salivary amylase, dilute mineral acids or heat. Dextrins form sticky solutions in water and are frequently used as adhesives, e.g. on postage stamps. They may have feeble reducing properties and when hydrolyzed, yield maltose and finally glucose. Inulin is a plant polysaccharide made up of fructose, soluble in warm water and does not give any color with iodine. Inulins are polymers consisting of fructose units that typically have a terminal glucose. Inulins have a sweet taste and are present in many vegetables and fruits, including onions, leeks, garlic, bananas, asparagus, chicory etc. Figure 1.18, inulin, N equals approx. 35, With this, we come to an end on our discussion on carbohydrates. In the following units, we will discuss about other proximate principles, proteins and lipids and their characteristic features. 1.7 Summary. In this unit, we got introduced to biochemistry as a discipline, its scope and relevance in context to dietetics. We started our discussion on carbohydrates, which form an essential part of our diet. This discussion, as you would now know, included definition and classification of carbohydrates along with their structure and properties. We learnt about various monosaccharides, disaccharides, their structure and chemical properties. We saw how we can distinguish these through different reactions. Finally, we discussed about various types of oligosaccharides and polysaccharides. Thank you. Subscribe to our channel for more updates, and we will see you with the next chapter.